Well, thank you for this invitation, and spasiba za vaš možnost. That's as good as I can do. I'm from the Complex Carbohydrate Research Center in Georgia. That's the other Georgia. In our group, we do computational chemistry, also synthesis, and biophysics, sometimes a lot of NMR, sometimes some crystallography. But the group is really interested in how carbohydrates interact with proteins. So today I want to talk about the development of drugs based on carbohydrates that are called glycomimetics. This is on the left a table of some glycomimetics that inhibit galectin. This is the protein shown on the center panel here. And you can see that the KD is significantly reduced as we have different functional groups added to the carbohydrate core. And we can go up to something like 2,000-fold enhancement of activity. And here's the crystal structure. You can see the central carbohydrate flanked by drug-like derivatives. The idea of using carbohydrates as a drug scaffold isn't immediately obvious because they're not very good drugs, right? They're hydrolyzed, they've got short half-lives, they don't cross membranes. However, they are very specific for their receptors. And so the idea is we want to exploit the natural specificity of the carbohydrate while changing it to enhance its affinity. Examples that exist as drugs, most people have heard of Relenza or Tamiflu for treating influenza. And back in 2009, John Magnani and Beat Ernst put together a very nice review. The problem, like all med chem problems, is really how do you choose these R groups? And many people will have a crystal structure of the carbohydrate in the protein binding site, but beyond that, it's really highly variable how you choose the groups. And as you can see in this structure, of course, there are conserved water molecules that are also something that needs to be considered. Our group is specifically interested in how pathogens infect cells, and this is the typical carbohydrate SNFG circles and squares representation of a sugar complex glycan. This image is actually of a cell membrane, and the cell membrane, the typical bilayer that we think of, is the thin black line running through this image. All this fur above it is called the glycocalyx. That's really a complex of glycosaminoglycans and glycolipids and glycoproteins. And this is the first thing that many pathogens see when they start to infect the cell. And because it's on the surface, the exterior surface of cells, many pathogens, including influenza, have evolved to recognize specific sugars in this forest of sugars. And that's what they adhere to as the first stage of an infection. And this is true for these viruses I've mentioned here. It's also true for things like several species of pneumococcus. So what we're interested in is, can we block this initial interaction with a drug that looks like this sugar? And the question is, how do you get there? If we zoom in on the binding site of influenza, this is the sialic acid, that purple diamond in the previous figure. And you can see that there's a pocket right nearby in the binding site. And one could imagine creating some sort of chemical moiety that you would attach to the sugar that would fit in the pocket, right? A very med chem vision. The difference with this approach is we're not screening a random library looking for hits. We're going to assume that the carbohydrate is the scaffold and the first primary hit. And then we want to do optimization. But how do you do that? We need to have a clever way to choose the R groups. Once we've done that, of course, we can run MD simulations to look at which one's a preferable inhibitor. These are the same influenza proteins, hemagglutinin proteins, but viewed from different angles. But this is one inhibitor, and we heat it up with water, and we run MD with a good force field, and the inhibitor just sits in the binding site. So presumably that's a good inhibitor. On the other hand, here's one that fits in the binding site by docking, but when you run an MD simulation, it dissociates and diffuses away. And so in principle, you can do things like this to help you choose which inhibitor you want to go forward with synthetically, and you can compute interaction energies from an MD simulation, as we've already heard today. But it still comes down to the fundamental problem of choosing the R group. Once our idea is fairly simple, if we had a library of R groups, we could splice them into the natural structure and then do some sort of conformational sampling in order to find a reasonable docked structure. So we're not really docking the carbohydrate back into the binding site. We're keeping the carbohydrate in the binding site from a crystal structure and just modifying the exocyclic groups. And we do this by performing initially a search of the dihedral angle space to find a plausible alignment and to get an initial interaction energy. And the first way we're doing our interaction energies is with autodock vena. So here's the overall view. What we want to do is create a web tool to do this, because then it can be open to a broader range of scientists. And the idea is they would upload a carbohydrate protein crystal structure, or plausibly it could be a structure from some other method. They would choose which site in the sugar they wanted to be modified, select the library of analogs that they want to screen. For example, they might want to screen esters or amides or things like that. At that point, we do the conformational search that I've just described, where we find the best pose. We can do some energy minimization on that and get plausible structures that we pass to MD simulation. So we're not just doing this with an implicit model, but once we've got a reasonable pose for the glycomimetic, we apply force field parameters and then run an MD. After that, we can do whatever analysis we want. For the library itself, we've designed, as I mentioned, to allow you to add moieties to hydroxyl groups or to amino groups, which are common in sugars. The moieties were scraped from chemical catalogs such as Sigma, or also from PubChem. Usually you get a SMILES string that we convert to 3D. And at the moment, I think we have, I could be underestimating this, we might be closer now to 1,000, but let's say 600 different moieties. These are not different molecules, these are different chemical moieties that you could put on at each site. So if we think combinatorially, if we've got 1, 2, 3, 4, 5 hydroxyl groups, that would be 600 times 600 times 600 times 600 different molecules. Once we've done the MD and we've got our structures back, we can calculate the interaction energy several different ways. Our group has done historically a lot of work with AMBER and MMGBSA or PBSA simulations for the interaction energy. We've also implemented a version of Autodock that we modified some years ago for carbohydrates. And now we're looking also at explicit water interaction energies. But as we do this, one of the things you discover in these structures, you often have aromatic rings. And of course, sugars have CH groups. And the CH groups in sugars interact with aromatic rings, and aromatic rings interact with CH groups in the protein. So we spent some time, and I'll talk a little bit about it, on the development of CH pi terms to add to the force field. What it will look like, and this is just a mock-up, but I really would like people to see where we're going with this. The concept is to provide a modeling platform that non-specialists can use, because if you can imagine, carbohydrates already are a very specialized area of research. And if on top of that, you require the carbohydrate chemist to be a computational chemist, there's 5 people in the world who are good at that, maybe 10. So that's not a very practical way to go forward. It's much better to have an online platform that has limited options and a robust interface that lets a non-specialist work with this sort of thing. So here we imagine they would upload a protein. On the next slide, they would pick the hydroxyl groups or the NH groups that they wanted to derivatize. If they wish to, if they know the kind of chemistry that they would like to use for the coupling, they can specify specific libraries or just all of them. And then they submit it. And there's a lot of stuff that goes on behind the scenes, because at this point we screen the libraries, we perform MD, we do the ranking, and you can rank them by multiple different methods. So if you find that when you compare to experiment, VINA is better than MMGBSA, you might choose VINA, or you might determine that MMGBSA is better for your scoring than VINA, so you choose MMGBSA. But what we return are the R group and the theoretical relative improvements based on the affinities. So I think this will be a very powerful tool. We built it not purely altruistically. I built it because it'll make it easier for our group to do this. But in doing this, it should be applicable and something that anybody can use, provided we have the resources to do it. Practically, what we have to do, we've got a library of R groups and a bunch of sugars. And we have force fields for the sugars. This is the glycam stuff. But the R groups, we don't have specific force fields for that. So we need some charges for each one, partial atomic charges. Then we need to add angle and bond terms, which we pick up from Amber's GAF, generalized force field, and we do force field mixing. Is this ideal? No, it would be nice to have accurate force field parameters for everything, but it's practical. Once you've got the ligand sorted out with the protein, since this is being uploaded from the PDB, there's all sorts of things that have to be done. You've got to remove the co-crystallization reagents, detect and form disulfide bonds, detect any missing segments in the protein. All these things that we manually do routinely need to be automated. And you can either repair those missing bits or place restraints on them. And then, of course, you take the ligand and you do the docking that I've just proposed. Once you've done this, from the MD, we extract representative structures. We do a binding energy calculation, perhaps an RMSD calculation, see whether it's drifting off, and then perform something like an MMGBSA or autodoc. And when you do this, sometimes you discover that there are missing terms that need to be in the force field, such as CH pi. If we look at a CH pi interaction for a minute, this is a system in sweet germ agglutinin, a lectin that binds sugars, specifically it binds N-acetylglucosamine. You can see the aromatic phenylalanine ring and the sugar placed above it. This is often called carbohydrate stacking. But what it fundamentally is driven by is two forces. One is desolvation, but the other is interactions between the CH groups that point approximately perpendicular to the ring face. And this is a CH pi interaction. In modeling, we basically model that traditionally as just a van der Waals interaction. But of course, a van der Waals interaction doesn't have any directionality. And it turns out it's not sufficient. Here we've done some QM calculations. This is with a reasonably high basis set. I'd have to check for details, but the patterns are all very similar. This is a canonical sort of view of what a CH pi interaction looks like. You've got the aromatic ring with a methane above it, with the CH bond pointing towards the ring. And if you vary this R group, you can go from no interaction energy down to about 1.5 kcal. And then, of course, it becomes repulsive as you push the methane onto the ring. Autodoc VINA does not capture this. It gets a very little piece of it, which is strictly just van der Waals in the VINA formulation. So you can also rotate the methyl into different orientations. You can look at what about if the methyl's over the bond or over an atom on the ring, and you can compute all these interaction energies. And you can also, in our paper, which I'll refer to later, we scanned horizontally across the surface in this canonical form as well. So we actually have 2D surface plots, and we did the best fit of our functional form to this, which is here in blue. As you'll note, one thing. The VENA repulsion is much less than the QM repulsion. And that's deliberate because in docking with VENA, you don't want to have hard van der Waals terms that reject plausible structures just because of minor collisions. So we kept our repulsion term roughly comparable to that of VENA. We also looked through the literature, well, not the literature, the crystallographic database. We looked through the PDB and found all instances of CH bonds that were on top of aromatic amino acids. And for example, you can see here is a phenylalanine ring, and these are all the CH bonds from different moieties that we culled from the PDB. You can see they're dispersed all over the ring surface. And if you look from above, you can see the same thing. We did this for phenylalanine, tryptophan, histidine, and tyrosine. The average of all of these is shown in this. So interestingly enough, the average is right over the center. And this is the ring centroid here shown as a purple sphere. And this holds true pretty much for all of the systems. So on average, it looks like the canonical structure, but at any instantaneous moment, it is not over the centroid. Our functional form, there's many different ways you could do this. We wanted to have a form that was dependent on the distance of the CH bond from the ring and on the angle of the CH bond, because if it's in a 90-degree, there can't be any orbital overlap, and so we decided that that was 0. So we use a cosine function to scale the interaction for an angular dependence, and we use a Gaussian function to determine the strength of the interaction, and we add that into VENA. We also chose, instead of having a function that was dependent on interactions with the centroid of the ring, that we would keep it atomistic. So our function is dependent on the interaction between the CH and each carbon atom around the ring. We were delighted to be invited to submit cover art, and this just happens to look kind of pretty, so I wanted to show it. But this is the 2D surface I mentioned, where we're scanning in this dimension. I believe it's the distance, and in this direction, this is the vertical distance, and this is the horizontal distance. So we looked at this in all docking software. You have to assign a weight to the new functional form. How much of it do you want to include in the docking? And so we looked at 2 parameters. One was we were doing blind docking of just a carbohydrate data set that we created. And we looked at it at 0. This is 0 means no CH pi term. 1 means 100% CH pi. And so you vary it and you monitor, for example, the top pose from docking is in green. At what value of the weighting factor do you get the highest percentage of acceptable structures? And the blue line is, let's say, any pose is acceptable within the top 5, and that sort of thing, and which ones are in the binding site. And you can see with no weighting factor, no CH pi term, 15% of the structures are acceptable. At a value of somewhere between 0.4 and 0.8, we sort of plateau with an improvement up to nearly 40% of the structures being acceptable. If we now look at the RMSD of the carbohydrate in the binding site, it also goes down. So this is comparing to the crystal structure. We can reduce it from, say, 20 angstrom RMSD, and this is blind docking, which I'll show why this is important in a minute, down to, say, 15 or 10, which is still a huge RMSD, but for blind docking, as I'll show you, it's really quite a significant improvement. And that's shown here. This is VENA docking a sugar to a protein. And we studied several systems, but this one's a representative. The protein surface is green, the binding site is red. When you apply VENA and you take the top 20 poses, you can see it, they really cover the entire surface with a few of them in the binding site. If, on the other hand, we apply CH pi term, we include that with a weighting factor of 0.8. You can see now we don't have any around the back or anywhere else, perhaps maybe 1, and all of them are generally in the binding site. but not exactly. So it's not a perfect improvement, but you can see it clearly brings the carbohydrates generally into the carbohydrate binding site. If we look at some examples then of MD simulations of these things, well, here's a mannose in the binding site of a protein called DC sign. We use this just as a control to make sure our MD simulations work well. We typically run these for about 100 nanoseconds. If the carbohydrate isn't stable on its own, then there's no reason to expect that your analog is going to work well. But if the carbohydrate is stable, then everything's fine. When you run the analog, now you can see this is a theoretical analog. We've got a phenyl ring here and some CH pi interactions here. But now this is different, right? Now we're running MD. We put the CH pi function into VINA, but we haven't put it into amber yet. So this is a simulation of classical amber with no CH pi. And although the aromatic ring stays sort of close to the valine, it certainly doesn't stay there the whole time. And fundamentally, the problem with this is that the MD force field doesn't have CH pi interactions. So whatever it captures is just due to van der Waals. If we now look at some correlations, this was just for that last system, DC sign. We plot the experimental binding free energy and the VINA relative binding free energy. We get an R squared of 0.52. It's not super exciting. It's not terrible, but you know, compared to MMGBSA, which is all over the place, in this particular case, VINA scored well. But there are many things to consider here. Many, many things. This is back calculated from the MD. If the MD doesn't have good CH pi in it, because it's lacking that term, you can't expect it to score all that well with VINA or MMGBSA. Also, we do experimental binding free energy measurements, and there's always a considerable concern that there's experimental error that isn't really captured when you're doing this sort of work. Everybody assumes it's a problem because of the scoring function or the computation, but it is important to remember that there can be significant variability in the experimental values. So hopefully I've left time for lots of discussion. Glycomimetic design, I think what we've shown is it is fully amenable to automation. And this is very powerful because everybody in my group who does computing has their own favorite way of doing it. But that really needs to be standardized. And that's what online web tools and automation ensure. They ensure that everything is consistent. I hope to have this online next year. That's where we're heading. There's plenty of scope for improvement. Parameterization of existing scoring functions is one area, but really what this helps us do, if you include known glycomimetics, it provides a very efficient way to create data sets that you can compare to experiment. Now, certainly we need to introduce some new physics. CH pi, for sure, that's not the only thing. Aromatic interactions or cation aromatic interactions, there's plenty of scope for improvement. How we handle water at the moment is terrible. We generally ignore it, or you use a generalized Born or some other function we'd really like to start using explicit water energy calculations. And of course, entropy is in the back of everybody's mind on these things. How do you best capture the entropy? We're currently looking at that, given that we have 100 nanosecond MD simulations. That's not enough to converge all entropies, but hopefully it'll be enough to help us to rank the energy contributions. And a big thing that we really have to do is, if we want to do MD simulations to improve our sampling, if it's sampling the wrong structures because it's missing some physics, then we have to correct that. And we're always looking for people who have carbohydrate glycomimetic data sets so that we can include those for comparison. So to conclude, I want to acknowledge we have folks who are doing all the background code development, because you can imagine in this automation process, you've got to go from a web interface that is nice and clean for the user, but behind that, you've got to do all the parameterization, the screening, the MD simulations, the data analysis, and then return it in a nice way to the user. And then the people who are working on the front end, they're, of course, always essential to this. And I've got to thank the NIH and the NSF for funding. And that should leave some time for discussion.
Thank you very much for the nice talk. Actually, there is a question in the chat. I'm sorry, it's not a question, but a comment. Nice work with very good theory practice balance. Thanks. And actually, I have a question dealing with your results when you compared, when you show the visualization, the cartoons of MD simulation and simulation in VINA. And you told that when you perform MD simulation, there are no parameters related to this pi-CH interaction. But, so how to say, well, still there is such interaction are observed, yes? And it's like van der Waals, well, effect of the van der Waals term. And, well, and why these van der Waals interactions are not, how to say, presented or are not working in the VINA? I didn't quite get this moment.
Well, they're not working in Amber either. The van der Waals is there. In Amber, it's a better van der Waals model than VINA. VINA deliberately has a soft van der Waals model because they don't want to. They don't want to have reject a lot of ligand poses in the docking. But in Amber, where it has a nice physics-based van der Waals, still only accounts for about 70% of the interaction energy. So that's comparing gas phase Amber to gas phase quantum. And
okay, so and this means that the minimum corresponding to this, well, the equilibrium is just lower than in VINA. I mean,
yes,
on the potential curve.
Right. So Amber is better, but it's not matching the QM. And when you run a simulation, for the most part, it's okay. But if you look, you'll see a lot more fluctuation of where there shouldn't be as much because it's missing the CH pi.
Thank you. And actually, we have a question from Roman Yefremov. And yeah, it's really a very important question. Thank you for the nice talk. And when including special new terms to force field, how to prevent double accounting of interaction? In this case, you will need to rescore all force field terms. This is a huge job. Yeah, actually, it's really an important thing.
I couldn't agree more, right? You have to be super careful with this. But it's a lot like when you fit a torsion term in a force field. You fit it as the difference between the observed result and the target results. So if you've got a QM curve that you're trying to reproduce, you're just fitting the difference between the observed and the target. So you're not just adding it on top.
Thank you. And actually one more comment and question. Thank you, Professor. At one of initial slides, pure sugars were shown. Do you design the molecules taking sulfur P interaction or not? Consider the option. Good luck.
Do I consider what? Sorry, I missed that.
Interaction between sulfur atom and pi system as I go.
Not
taking
yet.
Sulfur pi interaction.
Yeah, we don't consider sulfur pi interactions yet.
Okay, thank you.